Good afternoon. I'm James Hildreth. I'm Dean of the College of Biological Sciences, and I'd like to welcome you to the Storer Lecture today. I have the honor of introducing our speaker. Dr. Rice is the Maureen R. and Corinne P. Greenberg Chair in Virology and serves as the head of the Laboratory of Virology and Infectious Disease at Rockefeller University. He is one of the world's most accomplished virologists and is a prominent figure in research on members of the flavivirus family, particularly hepatitis C. He is an alum of UC Davis, got his bachelor's degree here in 1974, then obtained his PhD degree from Caltech in 1981. From 1986 to 2000, he was a faculty member at Washington University in St. Louis. His research team has helped to develop or understand the biogenesis and structure of HCV-encoded proteins. He's discovered a highly conserved RNA element in the 3' terminus of the HCV genome. And let me just say that it's hard to overstate the importance of Charlie's work in HCV. And that's been made more profound by his generosity in sharing reagents, highly specialized reagents to the rest of us who do research on viruses. That influence is even more profound by the number of students and postdocs he's trained over the years. So again, it's really hard to overstate how important his work has been for HCV. He's co-authored over 400 articles in virology. He's served as reviewers of numerous journals, is a former editor of Journal of Virology, and a past president of the American Society for Virology. He's a member of the National Academy of Sciences, a fellow of the American Association for the Advancement of Science, and has numerous other honors to his credit. But more importantly than that, he's just a genuinely prince of a human being, and I'm so happy to have him here to talk about his work. So welcome, Charlie. I'm happy to have you here. Thank you.
So we'll see if I'm on. Can people hear me? Yeah. Well, it's a great pleasure to be back at UC Davis. As Dean Hildreth said, I was an undergraduate here in the early 70s, so I guess that kind of dates me. Things have changed a lot, I have to say, although there are a few things that are the same. And it's actually kind of ironic to be giving the Storer Lectureship because in addition to sort of being an undergraduate here, I actually ended up getting my undergraduate degree in zoology. And sort of at the time that I graduated from Davis, I thought I was going to be a developmental biologist. I'd worked here on sort of sea urchin development, and when I sort of ended up at Caltech, the lab that I wanted to do my thesis work in didn't have room, so I got plopped in a virology lab. And that's really sort of the genesis of how I got interested in viruses. So this is kind of a circle in the sense that the Storers were great UCD supporters, and Tracy Storer was the sort of founding member of the zoology department. So I figured this would be a pretty diverse audience, so I wanted to give you really some highlights on hepatitis C. This, for me, has been a 25-year journey, which at times has seemed painfully slow in terms of our progress, but I think as I'll get to at the end, there's really been some remarkable progress on treating hepatitis C, and this, despite it taking 2.5 decades from the discovery of the virus, things are actually looking pretty good, although we still have some challenges ahead of us. So I thought, since this may be an audience that is, not thinking and breathing viruses all the time, what are hepatitis viruses? These are viruses that infect the liver and cause liver disease. It's kind of an alphabet of viruses. Hepatitis A virus, hepatitis B, and hepatitis delta, or HDV, hepatitis C, and HEV. These guys are sort of transmitted in different ways. HAV and HEV are transmitted via the sort of oral fecal route. And hepatitis B and hepatitis C, viruses that cause chronic infections, are transmitted by sort of blood or blood products. Now, in terms of our scorecard, and as I mentioned before, there's been a lot of work on these viruses over the years, and really quite a bit of progress. We have vaccines for the acute viruses HAV and HEV, also for hepatitis B. We have actually pretty good chemotherapy for hepatitis B, but I have to say that this is something that I'll sort of come back to at the end. Sort of despite having a vaccine for hepatitis B and good chemotherapy, we still have 250 million people that are infected with this virus that are really not necessarily receiving the treatment that they need. So there are really public health challenges in taking these advances forward. And for hepatitis C, we've really, the sort of driving force since the discovery of the virus has been to improve treatment for chronic HCV carriers. So a little bit of, sort of ancient history. Hepatitis goes back a long ways. There have been outbreaks of jaundice during many wars, dating back to sort of early stages of recorded history in 17th and 19th centuries. Outbreaks of serum hepatitis. Actually, there was an outbreak of serum hepatitis in horses called Tyler's disease that was sort of later recognized to correlate with transfer of serum, of antitoxin serum, autologous antitoxin serum in these animals. That virus is actually, or at least a virus that causes that, has just recently been identified. In the 40s, there were a series of outbreaks after vaccination for measles and yellow fever, and those were products that were often stabilized by including human serum. And it was recognized around that time that there were two types of transmissible hepatitis, one that was infectious, and these are the sort of waterborne fecal oral route viruses like HAV, and sort of serum transmitted virus, which later turned out to be largely due to HBV. And it was Baruch Blumberg in the mid-60s that sort of discovered hepatitis B virus in patient blood. This was visualized by electron microscopy a few years later. Hepatitis A was discovered in 1973, again by immunoelectron microscopy. This shows Bob Purcell, Al Kapikian, and Steve Feinstein, who were involved in that discovery. And it was recognized in the early to mid-70s that hepatitis A and hepatitis B really didn't account for all of the sort of post-transfusion cases of hepatitis. And this was recognized by a very talented and observant physician, Harvey Alter, at the NIH. And he termed this non-A, non-B hepatitis. And it occurred in about 10% of his transfusion patients. It often resulted in this persistent hepatitis, which could develop into these more severe outcomes, cirrhosis and cancer. And several groups later on showed that this could be transmitted to chimpanzees with human serum or blood products. So at that time, there was really, there was material available, but there was no reliable sort of antigen method to sort of screen for this, no cell culture system, and no molecular handle really to sort of get an idea of sort of what the causative agent might be. And so this initiated a race with many groups to try and identify the agent or agents that were responsible for this non-A, non-B post-transfusion hepatitis. And the winner was a group at Chiron led by Michael Houghton. He's shown here. And this was basically accomplished by using a bacterial cDNA expression library, a lambda GT11 library from convalescent patient serum, which was used for the screen, and they identified a single cDNA clone which they could then build up into sort of a near full-length genome of the virus. And this really sort of initiated the sort of modern era of hepatitis C research. They called this virus hepatitis C. At that time, I was a much younger assistant professor at Washington University in St. Louis and had been working on another virus, yellow fever. which it's turned out to be the prototype member of the family that hepatitis C ended up being in the Flaviviridae. And this was sort of an area that we began to study as a side project, which over the years developed into our sort of major research emphasis for 2 decades. So shortly after Michael Houghton and his group's discovery, there's a lot of sequencing done. It turns out that this virus is in fact responsible for the majority of cases of non-A, non-B hepatitis, the vast majority. There are 6 major genotypes. It's distributed throughout the world, and there are approaching 200 million people that have been infected with hepatitis C virus. So this is a real large problem. Now, the other problem from when you think about producing antivirals and vaccines against something like hepatitis C is this virus is amazingly genetically diverse. And this phylogenetic tree, the branch lengths, give you an idea of how different these HCV genotypes are relative to hepatitis B, for example, or clade B of HIV. So there's tremendous viral diversity. The idea that you could actually come up with new therapies that could be used to sort of combat the swarm of viruses around the world was a daunting task. And I should also say that in a single chronically infected individual, there's an estimated 10 to the 12th variants of hepatitis C produced every day. So really sort of all possible single and double mutations are sort of generated in this virus population in a single individual every day. So quite daunting. So a little bit about the natural history of this virus, which is kind of interesting. So I show here on this slide a couple of examples of a resolved infection and a chronic infection. And in blue, this is the level of circulating viral RNA, as you can see. And there's actually a very rapid rise in HCV RNA after acute infection, up to about 1 million copies per mil. And then you can see in red here, this is alanine aminotransferase. So this is a marker of liver damage. And this actually doesn't manifest itself typically in HCV until 2 to 3 months after infection. And it correlates with when the adaptive immune response actually kicks in, and probably starts to elicit inflammation and damage in the liver. And you can see 2 outcomes here. The one that we would like to see in every case, a resolved infection where the immune system is able to eliminate the virus, and a chronic infection shown here where there is some immune control typically that sort of lowers the viral set point, but the virus can, using mechanisms that we still don't fully understand, can sort of persist in the face of what is an otherwise normal immune system. So this is really the disease problem and the sort of kinetics of the disease problem, which I think are a couple of the reasons why HCV didn't receive initially the attention that it probably should have as a global health problem. So the first thing that you noticed here is that an acute infection can resolve, but that only happens in about 30% of the people that are infected with hepatitis C. About 70% go on to chronic infection. They can't control the virus. This acute infection, whether it's resolved or goes chronic, is typically subclinical. So people that are infected with this virus typically would not go to their doctor to figure out, what was causing their illness. The chronic infection lasts, basically for the lifetime of an infected individual, if not treated. And really the more severe sequelae, scarring of the liver, which can happen at various stages during this long chronic infection, or the development of cirrhosis, takes typically 2 decades or more to occur. That's not always the case. Some people can live with this virus for 50 years with very minimal liver damage, and other people sort of progress along this course to cirrhosis, which is very strongly associated then with the development of liver cancer. And this is still in countries where transplantation is practiced. This is the sort of major indication for liver transplantation, is basically end-stage liver disease, cirrhosis and liver failure. So it's a slow progressive disease. In the U.S., back in 2007, the age-adjusted mortality for hepatitis C surpassed that of HIV in the United States. And this is partially a function of the increasing age of the HCV-infected population, but it's also a tribute to the work that's been done for HIV to control this and decrease mortality with antiretroviral therapy and prevention and so on. But again, it's a significant cause of mortality. We haven't really sort of seen the peak of this yet because there are a lot of people that are infected with this, where it's been percolating for 2 decades, and it's predicted that decompensated cirrhosis or liver cancer is not going to peak until 2020 if sort of interventions don't change. And so really it's kind of a ticking time bomb, and this is just an example in the United States where we have 3.5 million people that are infected with hepatitis C. Globally, 170 million people infected with hepatitis C. So it is potentially a problem that is growing in severity, even though the incidence of infection is now relatively low. Shortly after the discovery of the virus, diagnostics were developed so that the blood supply, at least in developed countries, is essentially safe. Your chances of getting this today in the U.S. from a transfusion or a blood product are essentially zero. So the goals of research are summarized here. Clean up the blood supply. That's done. I wish it were done globally. I mean, there are countries that really don't have very good sort of surveillance and control on blood products. We take that for granted. High-risk groups, there's still the sort of frequent transmission of HCV in groups of young injection drug users, sharing equipment and so on. Education and needle share programs, I think, are helping. There's still the discussion of whether or not we need a vaccine. And then, in terms of treatment, there are a couple of challenges. One is we have to identify the people that are infected and then improve treatment. And I'll talk a little bit more about treatment and sort of how far we've come on that at the end of the talk. But the ultimate goal is really to try and put everything in place to try and eradicate this virus. So like many problems in human health, this one was not an easy one to tackle. And part of the reason is that these hepatitis viruses are incredibly difficult to work with in the laboratory. And it's been a bear to get hepatitis C to sort of replicate efficiently in cell culture. And I'll talk a little bit more about that. And there's really been no small animal model that's a good model for hepatitis C infection. And essentially all of the really pivotal work on the sort of discovery and early characterization of human hepatitis viruses was done with the chimpanzee model. And that model is no longer available for most kinds of research today. So a little bit about the timeline of research sort of beginning from the discovery and reporting of the virus in 1989. It wasn't until 1997 where we actually could produce a functional molecular clone for hepatitis C and show that it was infectious in the chimpanzee model. It actually told us that that was really all there was to initiating infection and sort of causing something that mimicked human disease. It wasn't until 1999, 10 years after the discovery of this virus, that Ralph Bartenschlager and Volker Lohmann developed what we call the replicon system. And this is a system where HCV RNA amplification, this is an RNA virus, could persistently occur in a human hepatoma line. So I think at that time, we felt that having a functional molecular clone, these replicon systems, which allowed cell-based screening and evaluation to occur, we were really sort of on the road to having a complete system. But none of these advances actually allowed us to develop a system that recapitulated the entire life cycle of the virus. And that didn't happen until 15 years after the reporting of this virus in 2005. And this was done really as a consequence of a really fortunate finding by Takashi Wakita in Japan, who had been studying a rare case of fulminant hepatitis after HCV infection, an isolate called Japanese fulminant hepatitis 1. And this isolate, or sort of chimeric viruses that were based on this, had the interesting property that unlike other primary isolates of HCV, it did not require adaptive mutations in order to replicate in cell culture. And it also had the remarkable ability to not only initiate replication and replicate efficiently, but it made functional virus particles. So this was really quite a boon. We could show that these were infectious in cell culture, they were infectious in the chimpanzee model, and they were infectious in a humanized mouse model that basically had human hepatocytes, liver cells, engrafted and expanded in a murine liver environment. So really a long road to really having all the tools that a molecular virologist would like to have to study a virus like this. I'll talk a little bit about primary hepatocytes and some of our efforts to establish small animal models for hepatitis C later. So I wanted to give you a few snippets of what we've learned about the virus life cycle. Being a molecular virologist, I figured I'd torture the audience a little bit with this. So this is our picture of the virus particle. You can probably tell that this is not a high-resolution structure of the virus particle. It's got two glycoproteins, a lipid bilayer envelope, a basic nucleocapsid, and a single-stranded genome RNA that's 9.6 kb in length. It's associated with serum lipoproteins, and in fact, ApoE is actually a structural component of the HCV virus particle, so antibodies against ApoE will neutralize HCV infectivity. It's got very heterogeneous buoyant density, and particles that have a lower buoyant density down here tend to be more highly infectious, maybe by association with more of these low-density lipoproteins. And the particles, even purified from this cell culture model that I described before, are really quite heterogeneous, and we haven't been able to get homogeneous enough particles to really get a good cryo-EM reconstruction of the virus particle. Now, this is largely a liver-tropic virus, and one of the questions that people have had is, sort of, how does HCV sort of choose the hepatocyte as a target cell? And this turns out to be actually quite a complicated process that I think we've not completely solved yet. But if we sort of look at this schematic, and these guys are hepatocytes, there seem to be low affinity interactions that are associated with interaction of the virus particle with glucosaminoglycans and the LDL receptor. And then there are two molecules, scavenger receptor type B1, which is involved in cholesterol uptake, and CD81, the tetraspanin, which is really present on all nucleated cells, actually bind directly to the E2 envelope glycoprotein of hepatitis C. We discovered a couple of molecules at the tight junction, Claudin-1 and occludin. Claudin-1 is enriched in the liver. These are both required for HCV entry. The sort of mechanism by which they participate in that is not completely clear. And there are other molecules, including growth factor receptors and this Niemann-Pick C1-like receptor protein, which is involved in intracellular cholesterol uptake, that also seem to be involved in HCV entry. So if we build up a cartoon of how this might happen, and in terms of a blood-borne virus making its way to the liver, the particle sort of sneaks through these sort of breaks in the endothelium that the liver has, interacts with these proteins on the basolateral surface, and we hypothesize triggers some signaling events which cause the virus particle to move to the tight junction where it's taken up by clathrin-mediated endocytosis. So that's at least the proposed entry pathway that people have elucidated by studying at least non-polarized cells. One of the things that we need to do now is to really sort of understand this in a polarized cell environment. So once the sort of genome is taken up, there is a sort of low pH dependent fusion step that the glycoprotein sort of delivers the RNA to the cytosol of the cell. And that liberates this 9.6 kilobase long RNA, which, as shown here, really has some atypical features for messenger RNAs. It doesn't have a 5' cap, doesn't have a poly A tail. It instead has an internal ribosome entry site that is near and dear to some in this audience. And this promotes direct 40S ribosomal subunit binding and initiation of translation of a long open reading frame that produces a protein of more than 3,000 amino acids. At the 3' end, there are structures and kissing interactions between stem loop structures that participate in HCV replication and potentially in translation in ways that we don't completely understand yet. One of the sort of interesting aspects of hepatitis C is that I think most of you are familiar with the burgeoning field of microRNAs. And usually these are associated with repression of translation or helping to sort of target cellular RNAs for degradation. In the case of hepatitis C, it actually requires two microRNA binding sites near the 5' end, miR-122, which is an abundant liver-abundant microRNA. And this probably explains at least some of the reason that this is a liver-tropic virus, because this microRNA is not found at high abundance in other tissues. So translation of this long polyprotein gives rise to 10 mature proteins that are produced by the action of cellular and viral proteases. These 3 proteins here that are shown in blue are the structural proteins that I mentioned before, core E1 and E2. There are 2 small non-structural proteins, P7 and NS2, that are involved in particle assembly. And then NS3, 4A, 4B, 5A, and 5B are really the guts of the RNA replication machinery. So NS3 has a protease which is responsible for cleaving between these components. It also has an RNA helicase activity. NS5B is the RNA-dependent RNA polymerase. So this is really sort of the engine, the generator of RNA replication. So it's this sort of core NS3 to 5B which forms the replication complex of the virus and sort of amplifies the genome through a complementary negative strand intermediate to make more positive strands. In terms of assembly and release, we know much less about hepatitis C in that regard. Obviously, the structural proteins are involved in these 2 small non-structural proteins that I mentioned. And there seems to be a close interaction between sort of the replication complex that is making new viral RNAs and the packaging components. So this is a tightly coupled process. So there actually is crosstalk between the replication machinery, probably delivering newly synthesized RNA to depots of core protein and other determinants that sort of target it for assembly of new infectious virus as opposed to entering back into the translation pool. And one of the key subcellular organelles that's involved in assembly is the lipid droplet. And this actually serves as a depot for accumulation of the viral core protein. And then the subsequent steps associated with, sort of maturation and budding of virus particles and secretion from cells, we really don't understand well. But it's believed that the glycoproteins are actually retained in the ER, and it's actually budding of and producing this envelope virus that mask ER retention signals and allow the virus to move through the secretory pathway where it becomes associated with serum lipoproteins. So that very quick sort of general overview of the life cycle of this virus is really a function of a rather limited experimental system. We were sort of fortunate, but handicapped, I think, in the early days of studying hepatitis C by the fact that all of the systems that I told you about that support RNA replication, that support virus production, are based on a human hepatoma cell line called the HU87 cells. And the bottom line is that hepatoma cells in cell culture are a lot different than hepatocytes in the normal liver. And one would really like to be able to sort of bridge this gap between the sort of contrived cell culture systems and actually what's happening in people. Now, I mentioned the chimpanzee model, which, as an animal model has been very important for many of the sort of seminal studies in these hepatitis viruses, including hepatitis C. The disadvantages are listed here. And sort of the recent decision of people sort of discussing whether or not it was ethical to use these animals for further hepatitis research has really made them pretty much off limits, except maybe for some aspects of exploration for HCV vaccines, although that remains to be seen. And so some of the efforts in the field have been directed at trying to understand more about how to maintain hepatocyte function and produce actually differentiated hepatocyte cultures that can be used to study these hepatitis viruses as well as small animal models. So I just wanted to give you a few sort of highlights of these efforts. So in terms of primary hepatocytes, the problem here, and I think this is a problem that most of us face regardless of what sort of pathogen we're studying, and that is sort of these viruses have, or pathogens in particular, have very specific host cell requirements in vivo. And when you try and take those cell types that might be their target cells in vivo and plate them in a dish, their properties change dramatically. And hepatocytes are just a classic example of this, where hepatocytes very quickly de-differentiate in cell culture to take on a more fibroblastic shape. They lose their potential to produce urea and albumin. And so one of the things that we've been doing is to really try and develop systems to sort of maintain hepatocytes in vitro and follow HCV infection. So before I get into that, let me tell you a little bit about a sort of system that we established to sort of watch virus infection in cells. And this isn't a big deal for virologists that, sort of study polio or things that, replicate well in cell culture. But I like showing this because, 15 years with really no virus that you could, study in cell culture, this was a big breakthrough for us. So basically what we did was to re-engineer a protein that the virus actually cleaves to inactivate the induction of type 1 interferon. So there's an adapter protein that's found on mitochondria called MAVS, IPS, or Cardiff, which is basically an adapter protein for the RIG-I pathogen sensing pathway. And hepatitis C doesn't like this adapter, and so it actually sends some of the serine protease that's responsible for polyprotein processing, and it chops it. And it cleaves off the adapter so it inactivates the ability of the cell to sense that the virus is infecting it and turn on interferon. So we basically modified this adapter to have a red fluorescent protein tag but retain the mitochondrial signal and cleavage site shown here, so that when we can transduce cells with a construct that expresses this, and then when we infect it with hepatitis C, it basically cleaves off this reporter, it relocates to the nucleus, and so we get this nuclear localized signal. So this allows us a very sensitive way to sort of look for a needle in a haystack in terms of a productively virus-infected cell. And this just shows you, this movie shows you an example of that. So we're looking at HCV antigen accumulation on the left, and these cells have been transduced with this reporter that I mentioned. So these are cells that are being infected, and you can see this focus of infection that is spreading over time. So I really love this movie. But anyway, so you can see antigen accumulation over here, and again, sort of virus spread, and we can use this to look at the effect of blocking agents or cell-to-cell spread and other parameters. So we've used this in a variety of different hepatocyte systems to see if we could find ones that support HCV replication. And this is an example where often cross-disciplinary investigations are really important. And I actually went to a symposium preceding the American Society for the Association of Liver Disease that was, it's established by a guy called Win Arias, who's really a fantastic liver biology guy. And there I met a tissue engineering person, Sangeeta Bhatia from MIT, who is really interested in systems to maintain hepatocyte differentiation, stimulate them to divide, basically with the goal of coming up with ways of making artificial livers to sort of help sort of salvage defective livers or people that had poisoning and so on while their livers would regenerate. And they developed this system called this micropattern co-culture system, where basically you can put little dots of collagen on a plate and pattern hepatocytes in those cultures, and then surround these with non-parenchymal cells, which basically, when optimized, particular ones work and others don't, those supply the diffusible signals and the contacts that are necessary to maintain these hepatocytes in their differentiated state for weeks in culture. And we could show that those could be transduced with this reporter, and they were permissive for HCV infection. So this actually allows us to begin to look at virus infection in a more authentic hepatocyte environment. But one of the drawbacks of this kind of system is that you need adult hepatocytes in order to make these micropattern co-cultures. And people are not often stepping up to the plate to give you a big chunk of their liver to make adult hepatocytes. So often I think with these things you can buy cryopreserved lots of commercially available hepatocytes at great expense, but you may run the risk of sort of running out of the cells that you're doing your study with before you've done all the experiments that you want to do. So I think one of the areas that is going to be increasingly important for infectious disease research is the sort of stem cell technologies to take stem cells or iPS cells and really sort of differentiate those into different cell lineages that are permissive for virus infection X, Y, or Z, And we've actually been able to do that, again, in collaboration with Sangeeta's group, to take iPS cells, differentiate them to hepatocyte-like cells, and show that these can be productively infected with hepatitis C. And this is really allows us a renewable source of cells, basically an infinite source of cells to set up and do these experiments. These iPS cells can be manipulated genetically using CRISPR and other genome engineering technologies to make knockouts or knock-ins of whatever you want. So I think this is really going to turn out to be releasing a big sort of bottleneck that we've had in this field. And you can imagine taking the same iPS lines and differentiating them into neurons or muscle or other cell types for other kinds of viral infections. So I wanted to say a little bit about trying to develop an alternative model for hepatitis C, and this is something that we've been working on now for about 7 or 8 years. So there are basically 3 strategies that you can think of. I'm sort of focusing here on maybe trying to make an HCV permissive mouse. One is that you can try and adapt the virus to, from a human environment to a murine environment. That's a pretty big jump. But that's one of the strategies that we've been taking. Another is to, as I sort of alluded to before, actually create humanized mice. In an ideal case, what we would really like is a humanized mouse that has a human liver and a human immune system, because really a lot of the pathology and pathogenesis associated with hepatitis C is probably immune mediated. So you want to have the immune system component. This is really difficult to do because there are so many different sort of cross-species signals that don't work sort of between mouse and humans, and you're trying to engraft sort of functional human components, whether they be liver or immune stem cells, into a massively immunodeficient mouse background that will tolerate these. So we actually have succeeded in doing this, but I guess the bottom line is that the sort of immune system function in these humanized mice is really not what we would like in terms of T-cell responses and antibody responses. So I'm going to tell you some of our progress on sort of genetic humanization, and there the idea is, well, what factors in humans might be missing in mice, and could we sort of engineer those into a mouse? Or are there incompatibilities between the virus and the murine environment that sort of preclude efficient replication? So in terms of thinking about this, why doesn't HCV replicate in mice? And it's true that from several studies in the literature over the years that RNA replication could occur in some murine hepatoma lines. So there wasn't an absolute, missing host factor that was required for RNA replication. Ralph Bartenschlager's lab in Germany showed that assembly and virus release could occur. But in terms of even just sort of getting an infection model, we really couldn't even get to first base in the sense that HCV glycoproteins could not mediate productive entry into murine cells. So that elicited a sort of number of efforts in our lab to try and identify the sort of human factors that were required to allow HCV entry into murine cells. And one of these tight junction proteins that I mentioned before, occludin, turned out to be sort of the missing piece. Sort of the human determinants in occludin, which varied in the mouse, were really what you needed to get HCV entry. CD81 was another factor that we'd actually known about for some time, that the murine ortholog of this protein didn't work very well for HCV entry. So there is a lot of work that sort of went into sort of discovering these kinds of things in cell culture, and then the question is, well, can you actually translate that into making a functional mouse model? And it actually works. So I'm leaving out some of the earlier parts of the story, but if you make a transgenic mouse that expresses either all 4 of these entry factors or just CD81 and occludin, the 2 human-specific ones that we need, this is actually an in vivo luciferase readout. So you can see that these mice are actually permissive for HCV entry. What I'm not showing you here is that this is actually a fairly sensitive system to measure HCV entry. In other words, we've actually re-engineered HCV to express Cre recombinase. And in the liver of this mice, they actually have a gene, a luciferase gene, that has a stop cassette where Cre recombinase has to be excised in order for luciferase expression to get turned on. So that's what we're looking at here. And so the good news was that these particles could get in and sort of express Cre recombinase. The bad news was that we didn't really see persistent replication. The sort of HCV RNA disappeared very quickly. I guess one of the things that I didn't tell you that we had noticed in some of the cell culture experiments is that really the innate immune response has an important sort of regulatory role in limiting HCV infection. And so one of the things that we thought worth trying in the mouse model was to just knock out some of these pathways that affect sort of the interferon system and innate immune activation. This sounds like an easy thing to do, but it led to an extremely large animal bill that we had to sort of foot for some years to generate these mice. And I'm just showing you some of the data here, but if you basically make mice that are defective in the JAK-STAT pathway, these are STAT1 knockouts, which is the pathway that mediates the interferon signaling effects to turn on a spectrum of ISGs, or IRF1, interferon regulatory factor 1. These animals were then not only permissive for HCV entry, but also for fairly long-term replication over 2 to 3 months. And we could show that in the serum of these animals there was actually infectious virus produced. So this is really sort of an example where actually 1 or 2 people in the lab that were really sort of driven to try and develop this model, which I felt had a very small chance of success, over years, really managed to actually do it. And so now one of the things that Alex Ploss, really the sort of postdoc that drive this, is doing is to really try and optimize this model and see if persistent infection can be established either by adapting the virus further to the murine environment, putting this in a more immunocompetent background, and see if some of the same pathologies that are associated with human HCV infection sort of develop in this model. So I wanted to sort of wrap this up by saying something about treatment, because that's kind of the sort of goal of all of our work in hepatitis C. And I think that despite the challenges, there are certain positives that I know that many in the room will envy, particularly those sort of working on HIV, for example. I mean, this is a cytoplasmic virus, so we don't have integrated copies of the HCV genome in some sort of stable repository. We believe that, and I'll show you some of the treatment paradigms that have been used, that successful treatment for HCV really does constitute a cure. In many cases, 99% or so, the virus does not come back after a successful treatment. There may be some residual RNA that you could amplify with very sensitive nested RT-PCR, but it's not clear that that is functional virus. And I think we're seeing with more experience with successful treatment of patients that if you get rid of the virus, it lowers your risks of liver disease. Not necessarily a sort of a surprise, but it's important, I think, when one is trying to decide who to treat and how much to pay for treatment, that we actually show that there's a benefit. So this shows you the sort of evolution of treatment over pretty much the time that sort of I've been sharing with you so far. So even before the virus was discovered, people had been using type 1 interferon, interferon alpha, to treat hepatitis B. So this is one of the first therapies that was attempted for non-A, non-B hepatitis, and this led to a pretty abysmal SVR rate, sustained virologic response rate, which is being virus undetectable 6 months after cessation of treatment, 6%, not very good. The next step was increasing the treatment length, which improved things somewhat, and then actually producing a biologically stabilized version of type 1 interferon by pegylating it, which actually improved sort of the treatment outcome for reasons that are actually, we could talk about this if there are questions, that are not completely clear. People were sort of treating interferon as a small molecule where you wanted to sort of increase bioavailability. And on the flip side of that, it's known that type 1 interferons are very tightly regulated, so that chronic administration of type 1 interferon on a cell actually turns off the interferon response. So it's actually not intuitively, doesn't intuitively make sense that more continuous would necessarily be better, but it was. The sort of next step in treatment was the addition of ribavirin. So this is a nucleoside analog, one of the few sort of broad spectrum antivirals that has been tried against a number of viruses. Its mechanism of action, sort of depending upon the indication, is still hotly debated today. It is an INPDH inhibitor, so it might actually decrease GTP pools. It also can be incorporated, so it might increase error frequency and push this already error-prone virus into sort of a catastrophic error-prone catastrophe. But this just gives you an example of where there's a lot of empirical science that goes into medical advances, because when ribavirin was tried as a monotherapy for HCV, it basically did nothing. There was like a transient half-log decrease in viremia, but really no marked antiviral effect. Combined with interferon, it actually gave rise to a 2 to 3-fold increase in this SBR rate for reasons that we still don't understand. So then the next step, PEG interferon and ribavirin, were up to about a 50% cure rate. And this regimen actually was quite effective for some of the HCV genotypes, genotype 2 and 3, but not nearly as effective for genotype 1, which is the most prevalent genotype in the US, Europe, and Japan. So that's where we stood in 2010. And then this is sort of where we entered the era of direct-acting antivirals. So these are the sort of 3 poster children of HCV treatment. 3 non-structural proteins that compose part of the replication machinery of the virus. Proteins that we and others have worked on. So the first one, excuse me, hopefully I won't lose my voice here near the conclusions. So the first one is the NS348 protease. As I mentioned, this is this guy that antagonizes innate immunity and is also responsible for processing. The crystal structure of that was determined back in 1997. The other one is NS5B, this RNA-dependent RNA polymerase, which, as I said, is the sort of engine that makes the newly synthesized RNAs. And then the third one is NS5A, which is a protein that's not only involved in RNA replication but also virus assembly. And this is a protein without any known enzymatic function, which was kind of a black sheep in terms of HCV drug development for some years because it didn't have enzymatic activity. People thought there was no way this was going to be a great target. It turns out that some of the compounds developed that target 5A are actually some of the most potent antivirals that have ever been developed. They work at sub-picomolar levels, sub-stoichiometric levels. Mechanism really not completely understood. And they have a very rapid effect not only on inhibiting RNA replication, but on the assembly of infectious virus. So in 2011, the first of these direct-acting antivirals was introduced and approved in combination with PEG interferon and ribavirin. These were 2 linear protease inhibitors, tenofovir and boceprevir. And on the one hand, this was great. It increased this SBR rate up to 70%. But these were very sort of genotype 1B targeted, so they had very limited genotype coverage. And resistance was a problem, and significant expense on top of the sort of interferon expense. And then side effects actually were really, I think, one of the reasons why people were hesitant to get treated for hepatitis C, because even with, you can imagine, high-dose interferon, which basically is associated with flu-like symptoms and depression for 24 or 48 weeks is not something to excuse me, look forward to. And ribavirin also accumulates in red blood cells, causes hemolytic anemia. And it turned out that these two linear protease inhibitors, tenofovir and bocephevir, actually just added more complications to this already rather poor side effect profile. So some of the people that had more advanced liver disease were treated with this triple combination therapy. About 20% of them actually ended up in the hospital, sort of needing transfusions and really pretty, close medical care and supervision. So this really created a situation where I think a lot of the physicians and patients were looking forward to, the DAA therapy, but, this really, was not what they were looking for. So a lot of them just sort of decided not going to get treated. I'll wait. So the ultimate goal has been to, eliminate interferon and come up with an all-oral regimen. I mean, interferon's an injectable. We'd like to come up with something that has pan-genotype specificity. I showed you some of the challenges associated with that at the beginning, given the incredible viral diversity, and something with minimal side effects. And this was envisioned to be a combination of small molecules that would target viral enzymes, potentially host targets. I won't have time to talk about those today, but there have been some of those that have been identified and have moved into sort of clinical development, and other direct-acting antiviral inhibitors like the 5A inhibitors. So this sort of gives you, I guess, in some respects, a snapshot of where all of the sort of basic science, biotechnology, and pharma have led us. And this is a schematic showing you a number of sort of the host targets that are shown in gray, but sort of the list of molecules in clinical development or approved that are protease inhibitors, 5A inhibitors, nucleosides or nucleotides targeting the polymerase, or non-nuc inhibitors. And you can see that there's really sort of a rich sort of pipeline of these compounds. And so I think that there's, even if we have some surprises during treatment, there are going to be a lot of alternatives. And I'll just show you, I think, a few slides that illustrate sort of where we are. And this is one that was published earlier this year, and it shows you some data with the limited number of patients. We have much more data on this now, of a 5A inhibitor, declatosphere, and sofosbuvir, which is a nucleotide prodrug polymerase inhibitor. And these are the SVR rates, either with just this two-compound combination alone or with ribavirin. And essentially, in this very sort of closely controlled, sort of clinical trial setting, we're looking at response rates, SVR rates that are better than 95% for some of these all-oral regimens. And there are multiple combinations that sort of show this behavior. And just 2 weeks ago, Harvoni, I don't know where they come up with these names. It sounds to me like some sort of Italian pasta dish or something like that. But anyway, this is a sort of 1 pill a day 5A inhibitor with sofosbuvir from Gilead that was approved just a couple weeks ago, which has these kinds of SVR rates, particularly for genotype 1. So really, quite a sort of revolution from these sort of 5% rates with 24 weeks of mega doses of type 1 interferon to an all-oral regimen, which it turns out, and I'll sort of comment on this a little bit later, is that some of these nucleotide prodrugs actually have extremely good cross-genotype coverage, and they have a very high barrier to resistance, so that we can select for resistance mutations in the laboratory under selection, but these very rarely appear in vivo because these resistant variants are so unfit that they really just can't seem to survive in vivo. So in terms of treatment, after this slog, we've got interferon-free combinations that are showing greater than 95% efficacy. There are multiple combinations showing promise. I think that's going to be an important thing to hopefully sort of drive down the sort of cost of these treatments, which I'll comment on lately. And there's really multiple regimens that have minimal side effects. These seem to work extremely well, even in the difficult to treat population. So historically, we've had a large population of HCV, HIV co-infected people. They have more rapidly progressive liver disease, and they're more difficult to treat with the interferon-based therapies. That seems to be falling away with some of these new all-oral regimens, and the co-infected population seems to be responding pretty much just as well as the HCV mono-affected. And I think for the patient and for the physician, these combination therapies are extremely well tolerated. They have very few side effects. Many of them can be used in combination with drugs that are associated with keeping HIV suppressed or immunosuppressive regimens in the sort of pre-transplant, post-transplant liver setting. So really quite a remarkable advance. So I think, the good news is that, we've started with this mystery virus in the 80s with, single-digit treatment to the discovery of the virus in 1989, 75% cure rates in 2011, with really sort of the capability to sort of get under at least ideal conditions to regimens that will treat by far the vast majority of people that get treated. So, we're sort of reaching the point where, is the game over? And I think HCV virologists are kind of asking this question, should we move on and do something else? And I'll talk a little bit more about that tomorrow. The answer is I don't think so. I think we should make sure that the problem is solved. And, I think we do have some real challenges. Some of those are scientific, some of those are public health challenges. And if we really want to get towards a world that doesn't have this virus anymore, we've got some real challenges. One is identifying the people that are infected. You can't treat people if they haven't been diagnosed. And even in the United States, more than half of the people with this virus that have not been diagnosed, they don't know they've got it. So, and that's actually a pretty good situation compared to other parts of the world. Implementation. We're not really all that good at implementing medical advances. And there are many examples of this as to how long it takes something which has been clinically validated as being better to achieving widespread use. It's not good. Even in the U.S., I think it's like a decade or something like that. In the case of HCV, we haven't done a lot better. So with 3.5 million people that are chronically infected with this virus, about half of them have been diagnosed and are aware. With all of the sort of summation of the treatments that have been used so far, 9% have achieved a sort of sustained virologic response. So hopefully with these new compounds, that's going to change because there's going to be, I think, less sort of fear of the side effects associated with the treatments. Affordable treatment. This is something that is, there's something in the paper or, a news wire every day on this. And this is a paper that Hill and Cook published, which basically shows you the cost of Sovaldi or sofosbuvir, same thing. It's $1,000 a pill for a 12-week course, $84,000. Samipivir, which is sort of a macrocyclic protease inhibitor, first approved in this class, pretty high, $66,000. Over here on the right, this is what the estimated manufacturing costs of these compounds. So you see we've got a gap that we need to sort of close here. And I guess one of the interesting aspects of this discussion, and Harvoni, the sort of combination pill that I mentioned that was approved a few weeks ago, is another 10 grand more expensive than Sovaldi. So I guess as a society we have to think about how we look at this. On the one hand, if you're Gilead that's making $1 billion a quarter on this, they would probably say, well, it's worth it. And if you really sort of say what's the effectiveness of this treatment at achieving an SVR per dollar, it's actually not so bad. Maybe better than the old days of interferon and so on and so forth. Well, I don't know. I think the insurers have other opinions as to sort of what they want to pay for these things. And I said there are multiple combinations that have these kinds of success rates. So I think there is going to be some market adjustment over time. And there have been some, I think, some good signs from the sort of pharma companies in terms of pricing. So in Egypt, where the prevalence rate of HCV is 10% of the population, they're going to make it possible to discount these compounds by like 99%. There are movement to sort of license the sort of technology and ability to manufacture these to generic companies in India and other places, and then they'll be able to sort of sell those and sort of give royalties back to the sort of holder of the license at a greatly reduced price. How the American and the European public is going to view this when we're paying this price, maybe it makes sense to just go see the pyramids or something, but I don't know. It's an interesting situation in terms of global health. The challenging patient groups, I mentioned, that looks actually very good. So even people with advanced fibrosis and cirrhosis are responding very well to these all-oral regimens. Not as good as the people with less severe liver disease, but really quite good. I guess one of the things that as a virologist, I kind of assumed that, liver has amazing regenerative potential. You get rid of the virus, everything is hunky-dory. The liver returns back to sort of a normal state. Unfortunately, that is not always the case. I think we have seen examples where people that achieve an SVR show remarkably improved sort of liver histology over time after they've achieved that. But once people have progressed to sort of more severe sequelae, particularly cirrhosis, there's still sort of a ticking time bomb for development of cirrhosis, and we don't have good biomarkers to really sort of tell who's at risk for developing cancer. So I think even when the virus is gone, we need to worry about sort of what are the processes that allow liver repair to sort of take place. How do we encourage them? How can we monitor for liver cancer, particularly in the patients with more advanced disease that get treated? And virus resistance, I think this is still potentially a problem. I mean, I've shown you that we're seeing these greater than 95% cure rates, and that is great, but one has to remember that these are under very ideal clinical trial settings at some of the best academic sort of research hospitals in the world. And sort of when these treatments get out into the real world, the patient populations are going to be different, the treaters are going to be different, and the response rates are going to be lower. And chances are there's going to be issues with compliance or forgetting to take these pills. There is going to be resistance, and as I've mentioned, I mean, our sort of viral sequence space out there is really daunting when you think about 170 million people infected, producing 10 to the 12th variants per day each person. That's a rather large number. And some of the classes of these drugs, other than the NS5B, nucleoside inhibitors, where they are efficacious, good resistance profile, and they have pan-genotype efficacy, some of these other guys are not so good. So, that is something that I think we will have to worry about. There are going to be treatment failures. We're going to have to sort of monitor resistance and figure out, the best way to retreat them. Hopefully, it won't be, nearly as much of a problem as it is in other chronic viral disease that are essentially sort of incurable or require usually sort of lifelong chemotherapy. And then what about a vaccine? This is something that we continue to discuss. It really has taken a back burner to sort of the small molecule development. And I think people are showing some resurgent effort and interest in developing vaccines. But, it has been one of these things that's lagging way behind. And we've had a problem with, sort of what is the economic driver of developing a vaccine. So, in the United States, if we have these therapeutic regimens where you can treat somebody for 12 weeks, maybe even 8 weeks, and get rid of the virus, are you really going to just want to vaccinate people even if they're extremely low risk? Even if they're at high risk, if they get infected, you could treat them. So, really sort of whether or not there's, the economic wherewithal, it kind of reminds me of the Ebola situation where, I think we have the wherewithal to sort of do the sort of preclinical development of, vaccine candidates for hepatitis C, but there's, not the sort of monetary interest to sort of drive it into, clinical trials except for a few that are going on now. And, that's a problem for a number of orphan diseases and also situations like this where a vaccine could be very useful in a country like Egypt, for example, where 10% of the population is infected. There's a very strong cutoff in the sort of age distribution of infection in Egypt, since this was probably propagated in an anti-schistosomiasis campaign that stopped in the early 80s. But what's striking is that when you have a viral load like that in the population, even in a country that would be considered to be fairly well developed, there's still transmission going on. So there's still, young Egyptians that are being born in that culture with, fairly significant HCV positivity by the time they're teenagers, probably by, bad medical practices or whatever. But so, in that situation, a vaccine could be useful. But I think, we've made some progress. I guess that's what I kind of wanted to get across in this talk. And it's really been a group effort from, very basic investigation in terms of, laboratory research to the biotech industry that sort of helped to develop some of these compounds, to pharma that can really, has the sort of wherewithal and the money to push the sort of clinical development of these. And I guess one thing that I would say, this does, I think, represent kind of an example of, basic science to what I hope will be a fairly successful translation. But I would say that, despite this being, showing this as a sort of arrow in this direction, a lot of our capability in terms of dealing like these situations, as in hepatitis C or other ones, has to do with maintaining, some level of a diverse research portfolio, often studying, viruses or pathogens or systems that have no obvious sort of medical, relevance, that later turn out to be incredibly medically relevant, whether it's either directly or it's basically trained a generation of scientists to do investigations in such a way that when, a new problem occurs on the horizon, they're sort of poised to do something about it. So with that, I'll just sort of show a few people that were involved in this. As I said, Alexander Ploss, who's now an assistant professor at Princeton, sort of really drove a lot of our sort of more recent animal model development. Sangeeta Bhatia and her lab at MIT, an incredible sort of tissue engineering group. I didn't talk a lot about kinds of the things that we're doing with her. Stephen Duncan, who's a stem cell biologist. But really, as I said, this has been, actually a great effort. And I think one of the things that's characterized the HCV field, which has been nice, is that people, despite being, competing to, discover the next receptor or whatever, it is a field where people have been very free about exchanging, the latest findings and reagents, and mutually supportive, which I think has sort of helped move the field along. So with that, I hope I haven't gone too long. I don't have a clock up here, but thanks everybody for coming and for having me here. It's been a real pleasure. So I don't know if we have questions or just start drinking immediately or no, get there. Yeah.
I was wondering if you could comment on the need for the vaccine a bit more, especially with regards to the diversity for the virus. I mean, if we simply implement it, for example, for everyone,
Well, I think it depends a lot on the country, and the sort of population. And, well, you actually sort of asked two questions. One is, sort of given the viral diversity, could we develop a vaccine or a cocktail that could, protect against all that diversity? I guess we don't know the answer to that, and I must say that sort of part of the vaccine pessimism in the past was the observation that people that resolve infection with hepatitis C or chimpanzees that resolve infection with hepatitis C can be reinfected. So in the resolution of a natural infection, you don't necessarily generate, protective or cross-protective immunity. I don't take the natural history of what happens with a virus in a host as sort of a predictor of our failure in terms of vaccination strategies, because I think these viruses do a lot of things to manipulate the immune response to, sort of gain the outcome that they want to, at least from kind of an anthropomorphic, sort of point of view. So I think with, different antigens, different adjuvants, maybe not focusing on, just a T-cell vaccine or just an antibody, neutralizing antibody vaccine. I actually think it's fairly low-hanging fruit. The problem is that without the chimpanzee, we really don't have a good preclinical model for testing. So, it's somewhat heretical, but I think for a lot of vaccination strategies, you'd like to be able to, there's so many different things that you could think about combining, whether they be immunization boosting with sort of heterologous antigens to get better cross-protective immunity to deal with that kind of diversity, or things that will elicit better T and B cell responses. You'd like to test these in people. And there's just a fairly high barrier to sort of getting the money that's necessary to get a clinical grade product to test in people. And I think if we could overcome that, maybe not that easy, then we could actually, do some human immunogenicity studies that would speed this along before one gets into sort of larger scale, immunogenicity trials. I was wondering, sort of speculating things have moved so fast with the, treatment response as to whether or not there would ever be a time or a significant enough database where you could actually do human challenge experiments. That in malaria, you can take a drug-resistant malaria and challenge people, so you can test vaccine strategies. Obviously, with HIV, you can't do that. But with HCV, with some of these regimens that seem to have nearly 100% efficacy with alternatives, I guess it's something worth thinking about. As I say, I think it's still potentially a long time in the future, but that would, if we could do that, it would certainly speed up vaccine development. So, yeah.
So many HCV infected individuals are also HIV infected, so co-infections have problem.
Yeah.
Do you think the HCV treatment for the AIDS cancer would it be something
Oh, I think so. I mean, you can comment on this better than I can, but I think in the sort of managed managed co-infected population, liver disease was a major problem, or is a major problem, and the ability to treat them with the same kinds of efficacies that we're seeing in the mono-infected patients, I think. Is going to sort of eliminate, HCV as kind of the opportunistic, comorbidity for HIV. At least I hope so. Was that your question? Yeah.
So based on those specific factors that you mentioned that seem to support the treatment, you would think that the virus would be a bit more promiscuous than what it is?
A bit more what? Yeah.
So I'd really like to hear your thoughts on why we don't see a broader spectrum of cells that's important.
Well, that's a leading question. There is actually a significant literature out there about HCV infection and other, cell types, B cells, for example, which don't have, some of the entry factors that we've come to believe in for hepatocytes. I think that, the literature is a little mixed in the sense that, when you find viral nucleic acid associated with the cell type, it's not necessarily evidence of productive replication in that compartment. And even if you can find negative strand RNA for HCV, there are some technical problems with the strand specificity of some of these negative strand assays that have been used. So I'm not ruling it out as a possibility, and in fact, it's something that, has concerned me, wondering that if this was really going to sort of rear an ugly head, sort of during these treatment regimens, because, if you look at the history of development of these small molecules, obviously the pharma companies are really trying to optimize these for the pharmacokinetics and delivery to, the liver, not the brain or the brain endothelium or other things. And I was, really quite concerned that, some of these more sort of liver-targeted regimens, might not eliminate potentially another reservoir like that. And, maybe that was sort of the secret of success of interferon, because it was, when it worked, it was sort of a systemic treatment. Cells, most cell types, essentially every cell type has a type 1 interferon receptor. So, but I guess the clinical data so far, with some of these combination regimens would say that, whatever, sort of tissues they're hitting, but, mainly the liver, is sufficient to, give rise to a durable SVR. So, I'm hoping that, we won't find, the virus in other cell types. I mean, it is an interesting question. And, one thing that we don't understand is, why does hepatitis C use all of these different entry factors to get into the liver? And how does it do it with, it's pretty efficient too. There are examples where chimpanzee infectivity experiments have been done with acute phase, HCV material, and shown that, it's not like this virus just sort of sticks willy-nilly to everything and doesn't make it to the liver. So the specific infectivity, even in that sort of, in vivo model, can be, like 10 particles are sufficient to initiate an infection. So, yeah, good question. I don't know, sort of all the answers. I guess one of the other sort of interesting aspects that I haven't talked about is that, for a long time we've wondered about what the origin of this virus is. Where did it come from, and, sort of. And we still don't know the answer to that. But with next generation sequencing technologies, hepatoviruses, there was one other closely related virus to hepatitis C called GB virus B. And it was sort of a single isolate. Wasn't really even clear where it came from, but it could infect tamarins. And never been seen again. And with next generation sequencing, we now have found that the closest relative to hepatitis C is not found in primates. It's actually a common virus in horses. So it's called we call it non-primate hepatovirus. It was originally identified in a respiratory outbreak in dogs, but then difficult to find in dogs again. But about 30% of the horses in herds, many herds are seroprevalent for this virus, and a significant number of them are chronically infected. Subsequent to that, it's been found in wild mice. Hepatovirus is still very distant relatives in bats. And sort of more recently, people have decided that they're going to get adventurous and have teamed up with the exterminators in New York City to examine the, sort of the subway rats and other vermin in New York, and found, hepatoviruses and other viruses in those rodent populations. So there's just all kinds of stuff out there, and we still don't really know where HCV came from. Some of those may not have the same, sort of liver restricted sort of tissue requirements that hepatitis C does. It might be transmitted by other routes. Yeah.
The difference in the chronic infection was that recurrent from an acute infection, would that be any indicative of the prospects of developing vaccine?
Yeah, I mean, certainly people have tried to look at that. And there are some inklings. You know, the initial studies were cross-sectional. And so one would find in chronically infected individuals, you know, very poor T-cell responses. And there would be, you know, antibodies that would neutralize the virus from a month ago, but not the currently circulating virus and so on, you know, sort of a la HIV. And then people did more longitudinal studies to sort of look for markers in the chimpanzee model to really try and figure out, you know, what are the predictors of, you know, spontaneous resolution. And we still don't really have a clear, you know, there's not a real clear sort of picture because sort of once you progress to this chronic state, there really does seem to be something active that the virus is doing to lead to sort of a stunned, you know, less functional, particularly a cellular immune response. One thing that is associated with both treatment response and spontaneous resolution is a genetic polymorphism in interferon lambdas. And so this was found in a genome-wide association study, and IL-28B, or interferon lambda 3, there was a SNP identified there which basically correlates with both interferon-based treatment response and also spontaneous resolution. We don't know sort of, you know, what's going on there. And sort of one of the paradoxes of that is the sort of bad SNP that's associated with poor treatment response actually has a co-signature of sort of elevated innate immune responses in the liver. So sort of like a pumped up sort of, you know, innate immune response in the liver presumably, you know, renders these patients that don't respond to interferon therapy just less responsive, you know, to the sort of exogenously applied interferon. So that's sort of a developing story on the lambda interferon front. But in terms of a smoking gun that would say, this is the, you know, this is the response that a vaccine needs to elicit to provide protection, we don't really have that, at least in the natural history of the infection. Sure. Okay, all right, well, good luck with the baby.